All right, welcome to this explainer. Today, we're taking a seriously deep, bottom-up journey into the serotonin system. This is one of the brain's primary diffuse modulatory systems, basically a massive network that starts from just this tiny cluster of neurons but casts a literal net of influence over almost every single part of your central nervous system. By the end of our time together, you're going to understand exactly how this fascinating chemical regulates our mood, our sleep, and honestly, some of our most complex behaviors. Let's get right into it. So here's our game plan for today. We'll start with making serotonin, look at the anatomy of the raffinuclei, talk about amplifying signals, dive into mood and antidepressants, and finally, wrap up with food, anger, and behavior.
Okay, part 1. Making serotonin, the chemical factory.
To truly get serotonin, also known as 5-HT, we actually have to start with your lunch, literally. It all begins with a dietary amino acid called tryptophan. Now, getting tryptophan into the brain, not easy. It has to fiercely compete with a bunch of other amino acids in your bloodstream just to cross the blood-brain barrier. But once it gets inside, this beautifully simple two-step manufacturing process kicks off. First up, an enzyme called tryptophan hydroxylase converts it into 5-HTP. Then, a second enzyme, 5-HTP decarboxylase, steps right in and quickly converts that 5-HTP into active 5-HT. Boom, you've got serotonin.
But here's the absolute crucial concept, the rate-limiting step. That first enzyme we just mentioned, tryptophan hydroxylase, acts as a massive bottleneck. You see, your brain's tryptophan levels are usually way too low to fully saturate this enzyme. So what does that actually mean for us? It means the overall speed of your serotonin production is strictly capped by how much tryptophan is available. In short, your diet directly dictates your brain's capacity to produce serotonin. How wild is that?
All right, moving on to section 2, anatomy, the raphe nuclei.
Now that we know how it's built, let's map out exactly where this system lives. Most serotonergic neurons are packed tightly into 9 structures called the raffinuclei. These are arranged right along the midline, basically the seam of your brainstem. And what's really fascinating is how they're split into 2 distinct territories. You've got the upper portion, the rostral group in the pons and midbrain, which fires axons way up into the cerebral cortex and limbic structures to manage things like mood and arousal. Meanwhile, the lower portion, the caudal group down in the medulla, shoots its axons downward into the spinal cord to modulate pain-related sensory signals. So it's the exact same chemical, but with vastly different destinations and jobs depending on where it originates.
Which brings us to section 3, amplifying signals in the brain.
So you might be wondering, how does such a small cluster of neurons influence the entire brain? Well, the secret sauce is in the receptors. Unlike fast, direct on-off switches, think of glutamate for example, serotonin uses G-protein-coupled or metatropic receptors. You really want to think of serotonin less like a light switch and more like a master volume dial. Instead of just sparking an electrical signal directly, it modifies the effectiveness of signals from other synapses. When it binds to its receptor, it triggers these massive biochemical second messenger cascades like the CAM pathway. Basically, a single molecule of serotonin can activate a whole cascade of downstream enzymes, leading to broad, sweeping, and super long-lasting changes in neuronal excitability. It's a huge multiplier effect.
Okay, let's shift gears to section 4, mood and antidepressants.
All this deep biological groundwork leads us right to the monoamine hypothesis of mood disorders. This is the idea that clinical depression stems from a literal deficit in this specific modulatory system. And the way we figured this out is a fascinating bit of history. Scientists started noticing stark patterns based on how different drugs interacted with serotonin. For instance, when patients were given rezapine, a drug that depletes serotonin by blocking it from loading into storage vesicles, they developed severe depression. But on the flip side, MAO inhibitors, which stop the enzymes that normally destroy serotonin, actually elevated mood. And imipramine, which blocks the brain from reabsorbing serotonin, turned out to be a highly potent antidepressant. The pattern was just undeniable. More available serotonin equaled a better mood.
And that historical breakthrough brings us right to the modern era of treatments, specifically SSRIs, or selective serotonin reuptake inhibitors, like Prozac. These drugs are designed with incredible precision to target the presynaptic membrane. By blocking the serotonin transporter, they basically stop the brain's cleanup crew from clearing away the serotonin too quickly. This brilliantly leaves a lot more serotonin just floating there in the synaptic cleft, prolonging its interaction with those G-protein-coupled receptors we talked about earlier.
But wait, this brings up a really fascinating mystery, what we call the paradox of delay. Think about it. If an SSRI drug boosts synaptic serotonin almost immediately upon taking the pill on day 1, why on earth does the patient usually not feel any emotional relief for several weeks? The answer to this completely changes how we view antidepressants. The emotional relief isn't just because there's more serotonin floating around. Actually, that initial serotonin spike is really just the trigger. Over several weeks, this sustained signaling prompts some incredibly deep, long-term adaptive changes in the brain's physical structure. We're talking literal alterations in gene expression, and, amazingly, it drives increased neurogenesis, the actual birth and growth of brand new neurons in the hippocampus. That physical, structural rebuilding of the brain is what takes weeks to happen, and that is what ultimately lifts the depression. It's not just a chemical band-aid, it's a structural remodel.
Finally, let's wrap things up with Section 5, Food, Anger, and Behavior.
Let's connect all this deep biochemistry right back to your daily life, starting with the classic food and mood connection. I mean, we've all felt that comforting boost after a huge carb-heavy meal, right? Well, here is the exact biological sequence behind that. When you eat a bunch of carbs, your body releases insulin. Now, that insulin actually sweeps away many of the competing amino acids from your bloodstream. Remember earlier when we said tryptophan struggles to cross the blood-brain barrier because of all the heavy competition? Well, insulin just kicked out all the competitors. So tryptophan absolutely floods into the brain. Serotonin production spikes, and your mood naturally goes up. It's a direct chemical reaction to your lunch.
But it gets deeper, because serotonin doesn't just reward us for eating, it's the signal that tells us when to stop. The concentration of serotonin in the hypothalamus acts as a highly sensitive biological gauge for hunger. It actually rises in anticipation of food and then spikes during a meal. High serotonin acts as a powerful satiety signal. It literally tells your brain, okay, we're full. Conversely, abnormally low levels in the hypothalamus are directly associated with an insatiable appetite and are heavily linked to severe eating disorders, like bulimia nervosa.
Serotonin's behavioral reach goes way beyond food, though. The serotonin deficiency hypothesis shows us that this system is deeply, deeply tied to how we handle anger. And the data here is striking. There's a strong inverse relationship where lower serotonin activity actually correlates with higher levels of anger and aggressive behavior. We know this because we can measure something called 5-HIAA, which is just the leftover metabolite of serotonin found in cerebral spinal fluid. Lower levels of this byproduct reliably, almost perfectly, map to higher aggression in humans.
Now, you might be thinking, if serotonin is this broad volume dial for the whole brain, how could we ever treat aggression without making someone completely lethargic or exhausted? The answer is receptor precision. See, the brain doesn't just have one type of serotonin receptor, it has at least 14 distinct subtypes. And scientists have pinpointed that 2 of these in particular, 5-HT1A and 5-HT1B, are specifically responsible for modulating aggression and anxiety. By designing drugs that act as targeted agonists, meaning they stimulate only those specific receptors, researchers can successfully calm aggressive behavior in animal models without shutting down the entire diffuse modulatory system.
So what you should really take away from this explainer is the sheer elegance and staggering complexity of the serotonin system. I mean, it starts with a simple amino acid from your food and cascades into the literal structural rebuilding of your brain. As we sit here today, mapping out the precise functions of all 14 of those receptor subtypes, it leaves us with a genuinely thrilling question. As we decode this puzzle, how much more control over our mental health will we gain in the near future? Could hyper-targeted agonists become the ultimate, side effect-free treatments for modern anxiety and aggression? Honestly, the closer we look at the biochemistry, the more hopeful the future of mental health becomes. Thank you so much for joining me on this deep dive. Keep learning, and I'll catch you next time.